Today we're learning all about the wonderful world of B cells and the antibodies that they produce. When we look at the third line of defense, it is a super specific, super targeted response. And it's thanks to antibodies that we are able to create a force against a pathogen that's able to wipe out the infection super quickly. The activation and release of antibodies is thanks to these cells called B cells, and these are another cell that we have in your third line of defense. We've looked a little bit at T cells already in a previous video, and we're going to be looking at B cells today, how they're activated and how they do actually produce antibodies. This is known as a humoral response, or the humoral immunity. In biology, humor just means like the fluids in your body, so it could be like your blood or it could be like your tissue fluid. So we're looking at something that's getting released out into these fluids, moving around the body, and then finding pathogens really quickly to stick on their surface. As antibodies are able to travel out in the fluids surrounding your cells, this is really important against extracellular threats. When we looked at T cells in that previous video, we were looking at cell-mediated immunity, which is cells killing cells that are faulty or intracellular threats. Extracellular threats are things like bacteria, free viruses, that sort of thing, and that's when an antibody will attack. Antibodies will often work in conjunction with the cytotoxic T cells that we have in the cell-mediated response. So if we consider a viral infection and we've got T cells killing cells that are infected with a virus, so the cytotoxic T cells coming in, triggering the death of those cells, that's fantastic for the intracellular threat. If the virus is being released out of the cell, however, those T cells are not able to kill that virus. So in that case, we're going to be relying on antibodies being produced, which will clump up the virus and kind of neutralize them. B cells take a little while to actually activate because we do need to go through like a lot of steps. We've got to get through the second line of defense. An antigen presenting cell needs to rip off the antigen. We need to find a T cell, and then that T cell tries to find a B cell. So to see a full immune response with humoral immunity, it might take about 10 to 17 days. B cells are a type of lymphocyte, so these are the types of cells that we tend to see in the third line of defense. They live in the lymphatic system, so in lymph nodes, lymph vessels, that sort of thing. With our B cells, they do originate from the bone marrow like all other lymphocytes, but they also mature in the bone marrow, unlike the T cells, which mature in your thymus. This is where we get the names from. The process of maturation is actually basically weeding out all these cells and getting rid of any self-reacting B cells or T cells. We don't want those because they're going to cause some problems later on, but that's a future video. In order to activate a B cell, we actually need the help of helper T cells. So if you want to have a look at how helper T cells actually get activated to begin with, watch our previous video on T cells. We'll just do a quick recap today. So if we've got an invasion, second line defense has been activated, our antigen presenting cells will have engulfed our pathogen and ripped off the antigens. These antigens are going to get presented on its MHC class 2 markers, or MHC2 for short. So let's focus on a macrophage today, one of the antigen presenting cells. It's going to carry the antigen from the pathogen that it's just eaten, and it's going to carry it onto MHC2. This macrophage is going to move into the lymphatic system next. The lymphatic system has all these little lymph nodes everywhere, and lymph nodes carry lots and lots of T cells and B cells. We're going to have our macrophage move through to the lymph nodes and basically try to find the one T cell to activate, the exact right one. We're looking for a complementary shape between the macrophage's MHC2 and the antigen that it's holding, which is our epitope, and we're looking for that to be complementary to a T cell with a T cell receptor. The T cell receptor needs to be complementary. If it's not complementary, we will not activate this T cell. So the process of matching up the MHC2 and the antigen and matching up that epitope with a T cell receptor, this is known as clonal selection. There are lots of T cells around. We need to find the exact right one. Once we find that T cell and we activate it, that T cell is going to undergo clonal expansion. So we're making lots and lots of copies of this T cell, which differentiate into helper T cells and all the other T cells that we have. So we'll just focus on the helper T cell. This helper T cell is a regulatory T cell. So it's going to be responsible for stimulating other cells in your immune system, so like phagocytes, cytotoxic cells, that sort of thing. But its job is also to find a B cell and activate that and stimulate that using cytokines. So when we have our helper T cell activated, we've got lots of them now, and they've got a complementary receptor with our antigen on it. They're going to go and find the right B cell now. That B cell could be anywhere in the body, so they need to move from lymph node to lymph node and find the one. We're going to find that B cell and we're going to bind to it and we're going to activate it. So it's another process of clonal selection. That T helper cell is going to move around, it finds the right B cell, and it is going to bind to that B cell's MHC2. So MHC2 is just found on our B cells and antigen presenting cells, and it's complementary again to that T cell receptor. So once we find that, we're going to bind that B cell and we're going to release out cytokines. They're little signaling molecules to tell us that this is the cell we want to activate. Once that B cell's received that signal to activate itself, it will clone itself into another B cell army. So we've got another round of clonal expansion. Lots and lots of mitosis of this naive B cell that happens to be the right shape. And we're going to make a big army of B cells which differentiate into two main types of B cells. We have our plasma B cells, which are responsible for releasing out antibodies they produce and release. We also have memory B cells, which are responsible for holding on information about this pathogen and staying in your system forever. This is responsible for long-term immunity. When you encounter this pathogen again, this memory B cell knows exactly what to do, and it will rapidly clone itself and make plasma B cells and deal with the threat before we realize. Plasma B cells are going to be responsible for producing a bunch of antibodies which get dumped out into the humors of your body, to the fluids, and it moves around and gets to the area of infection. These cells are going to be working really hard. It uses a lot of energy because they're constantly producing these proteins and releasing them out through a process called exocytosis. The production and release of antibodies is a really rapid but also short-term response. The production of these antibodies are using up a lot of energy, so we can't sustain this indefinitely. We'll have plasma B cells releasing antibodies as long as we've got that threat in our body. Once we're done, these B cells will die and the level of antibody that we have in our bodies will actually decrease. Because antibody levels do vary over time, this is actually a really good indicator of what stage of a disease you're in. So if you have an infection and you are in like the peak of the infection, your antibody levels will be really high. And if you are, dealing with the infection, everything's sort of like being taken care of, your antibodies levels will drop. It's really important that we know what an antibody is and its shape. When we consider an antibody, it is a protein. It's being produced by your plasma B cells and being released out. A protein is going to be a really small molecule that is made up of amino acids, and their shape is really important. So if an antibody is the wrong shape, it's not going to work against our pathogen antigen. An antibody has a very particular shape. It's vaguely Y-shaped, and it's got these things called antigen binding regions at its end. And this is the bit that's complementary to the antigen of our pathogen. So when we look at an antibody, it has heavy chains, which are the middle sections, and then it's got this light chain as well, hanging off the sides of the heavy chain. At the end of these regions, they are variable. So this is the bit of the antibody where we can have different shapes. And because we have different shapes, we can have antibodies that are specific to different antigens being produced. The variable binding region kind of is like the handcuff region. So it's a bit that's directly in contact with the antigen's epitope. The rest of the antibody, it is constant. So we have a really set shape amongst all antibodies. Antibodies produced by your plasma cells will just get released out. We have some antibodies that are also found on the surface of your memory B cells, and this is what makes that memory B cell so special. Our memory B cell is going to activate itself by having that antigen binding to the antibody on its surface, which is a really specific antibody, and then we have a triggering of different reactions within that memory B cell. So the next time you encounter a pathogen, that triggering is going to happen really quickly. That memory B cell will clone itself again, and we're going to have a massive immune response. Whilst antibodies are very specific against an antigen, they do have a relatively wide range of action. So some antibodies will bind to the surface of a pathogen on its antigen. It'll bind to these antigens and it'll tell nearby phagocytes to come in and eat up this particular pathogen. So by doing this, we also can neutralize the pathogen. So let's say if it's a virus, this virus can no longer get inside of other cells. And by triggering more phagocytosis, so signaling for phagocytes to come to the area and eat up this pathogen, we also know this as opsonization. We also have some antibodies which are responsible for recruiting things like cytotoxic T cells to an area and triggering the death of some pathogens. So if you've got an antibody bound to a particular cell, a T cell will come by, work out that this is actually an infected cell, and it'll trigger apoptosis of that cell. Antibodies can also trigger agglutination, which is essentially clumping. If you consider an antibody, they have two bits that can bind to a pathogen's antigens. So it's at the end of the Y-shaped regions, you have two binding areas. You can bind to one pathogen with one side and bind to another pathogen on the other side. You can form a complex with lots and lots of antibodies and lots of pathogens. And if you're a phagocyte, that's really easy to eat up. You can see that thing, go like, that's weird, I'm going to eat you. Agglutination is one of the reasons why it's really important to test for blood before you get a blood transfusion. Your red blood cells do contain antigens, so they might not have MHC1 because they don't have a nucleus, but they do produce antigens that get stuck onto the surface of the red blood cell. The antigens that we have are inherited from your parents, and it's inherited in a co-dominant fashion. So as long as you inherit one of those genes, you will definitely produce one of these proteins on the surface of your red blood cell. The different antigens that we have are either A, B, or D, which is also known as a rhesus factor. You will be a combination of A, B, or not having A, B, and you'll either have antigen D or rhesus factor, or you don't have it. So a combination of those give you your blood type. If someone has AB positive blood, what they have on their red blood cells is an A antigen, a B antigen, and the rhesus factor antigen. If someone has O negative blood, they have no AB antigens and they don't have the rhesus factor antigen. Because O negative blood is a universal donor, in cases of emergencies and someone needs to get a blood transfusion and we don't know their blood type, you would give them O negative blood. However, as much as possible, matching up the blood antigens is the best call. So do know your blood type. Also, when you're older, or if you are old enough, please do donate your blood because it is making sure that people out there are getting what they need to survive. We've actually got a few different types of antibodies that are produced by your plasma B cells. We can call antibodies immunoglobulins as well, because bio loves the vocab even more here. And if we look at the different types of antibodies, the immunoglobulin gets shortened to IG. So we've got IgG, we've got IgM, IgA, IgD, IgE, lots of them. I don't know what they all stand for, but they exist. IgMs are the antibody that you tend to see in the first immune response. So upon first exposure to a pathogen, we see more IgMs being produced, and they're really good at activating the complement protein system in the second line of defense. IgG is found mostly in secondary exposure to a pathogen, so they last for quite a while in your serum, and they're really good at activating phagocytosis by macrophages. IgA is found mostly in external secretions, so you might see this in breast milk, and it gives the young some immunity as long as they're breastfeeding, and you can also see it in your sweat. IgE is found in allergic responses mostly. They are really effective against parasites, but we tend to focus on IgE and the effect on allergic responses. Future video spoilers there. And IgD, we're not really sure. We're still researching this stuff. So IgD, we know that this is an antibody. We're thinking maybe it's got to do with receptors on B cells, but we're not really sure. So the world of antibodies and immunological research, we're still learning a lot. Releasing antibodies, it's a bit of a game changer in an infection. So once this is done, it's kind of game over for most infections. our antibody levels take a little while to rise. So if you see a graph like this, you often see that antibody levels in serum take a little while to actually get released because of that slow activation of your third line of defense. But once it does get activated, the antibody levels will increase to a certain point, and then as it decreases, this is a sign that the infection is over. This is a sign that we are winning against the pathogen and we're no longer requiring those plasma B cells to release as many antibodies. After an infection, the antibody levels in your serum, so this is your fluids, this will decrease quite a bit. So for some people, this never gets to zero. You have a little bit in your body still because you might have some lingering plasma B cells doing their job. But most of the times we kill all those immune cells that we had working for us because it's a lot of energy to keep them around. Looking at this graph, you can also see a secondary exposure. So this is us encountering the pathogen for the second time. It has to be the same pathogen with the same antigen. So once this happens, we are going to bind that antigen directly to that memory B cell, and it gets activated to make that clone army once more. That clone army differentiates into the plasma B cells as well as some more memory B cells, and we are producing a lot of antibodies super quickly. B cells and antibody production are the end game of your adaptive immune response. Your third line of defense, it's kind of nothing without antibodies. Like T cells, you're great and all, but what about extracellular threats? That's the majority of pathogens. B cells, they've got your back in that case. They're the ones that are producing a massive response against that pathogen. It's the equivalent of bringing a machine gun to a knife fight. You're definitely gonna win. As well as that, your B cells, we've got long-term immunity, we've got memory. So your memory B cells, they'll stay in your system forever. They're your ride or dies. They're gonna remember that pathogen and take care of it for you. So thanks to our memory B cells, thanks to all of our humoral response, you're able to survive today. You're able to be alive, to study biology in this crazy time, this crazy world that we have. So happy studying and good luck.